I'll just tell you what we're going to be doing this afternoon. Where are we? 1 o'clock. For about the first, 45 minutes? For about 45 minutes, Chris is going to go through an example, and we're going to get your input into this, into developing a surveillance system. So we're going to try and use a local disease and get your comments and your thoughts. So we're going to start a little bit like the group is a think tank. So we're going to start off with the disease and ask you questions and try and put something together over the next 45 minutes. After that, then I will take over and do another little exercise that's going to involve some group discussions and group work. Nothing too challenging, but just something that will hopefully get you stimulated and get you thinking about diseases in wildlife, zoonotic diseases in particular, but other diseases as well. So we'll start off with a little group exercise on one of the flip charts. Then we'll separate you into groups, and you'll have an hour together in groups of 3 to 5, depending on how many categories we come up with on the flip chart. And then you'll come back and you'll report on your findings. But we'll give you more instructions as we go along. So it'll be an afternoon where you may feel a little bit sleepy after lunch. I always feel a bit sleepy. So hopefully we'll give you something to wake you up a little bit later. So, thanks.
How many of you met her last year? Some of you? She told me to ask, I can't remember, it was either Paisan or Sai about the langur. Is that you? Okay, we have to talk about that later. So she sent her greetings to everyone, and she's sorry that she couldn't be here. She's hoping that she can come for the next one. I also wanted to clarify when the Took mentioned the tiger bomb yesterday, and since I was the only one that raised my hand, I just want to make sure that everybody understands that I haven't been trading in tiger parts or anything like that. This is a commercial product, and the time I've used it actually was in Africa to keep the bugs away. So I just wanted to make sure everybody knows that I haven't been using tiger bones or tiger penises or tiger testicles or anything. So, okay, this session is going to be a bit of an experiment, but we're going to try to. The feedback from you guys is that you want more sort of practical, applied, how to do things. So we're going to spend the afternoon trying to sort of go through some of those things. For this session, we're going to try to talk about how to develop a surveillance system, but in fairly general logistical terms, and not get too much into the details of sampling numbers and things like that. Hopefully we can get more into some of that with the individual groups later. But so as a reminder, as Dominic presented earlier, these are the basic elements to designing a surveillance system. So let's just imagine a scenario where you're the veterinarian in a national park, and you have a diagnosis of a Nipah outbreak in a human community surrounding the park where you're working. Okay, so just imagine that's something like that is happening. And then your boss comes to you and he wants to know what your plan is, because people are already starting to be scared. They don't want to come to your park. They're worried that the wildlife might have Nipah and that they're going to start to die. So this is the kind of scenario that you might be faced with as a wildlife veterinarian, and you have politicians or people above you that may not understand everything, but they're going to tell you that you're responsible for trying to figure out what to do. Okay? So I think hopefully this is the kind of exercise that will help us start to help you figure out ways to start developing surveillance programs like this. And that's not really the director of DNP. So, I should probably hide this. What do we know about Nipah virus? I assume you have all studied it very well. So, what can you all tell me? And do we have another microphone? What are the basics of the ecology or epidemiology of Nipah virus? This is where we're going to test who ate too much for lunch. So for the whole rest of the afternoon, you guys you'll have to participate. So we're not going to just sit up here and talk anymore. We need to.
No need to go into great detail. Just give some facts about Nipah virus.
What do we know? What does anybody know? Just raise your hand or call out something to stand. And feel free to speak in Thai, and somebody can translate.
The first import is in Malaysia.
Okay.
That's a great start.
What else?
It's an RNA virus.
Sorry?
It's an RNA virus.
RNA virus.
Virus?
What else? Associated with therapist bats. What else do we know?
Any other animals involved? Pigs? Any others? Bats?
What animals have I already mentioned?
What was the most important animal affected?
Humans. Don't forget, we are also animals.
Any other animals that you know about that were linked to the outbreak or at least involved? Another animal species that was loosely linked, certainly in the eye of the authorities. Dogs. Now, dogs probably aren't part of the epidemiology, but there was some suggestion that there might have been some seroconversion in dogs, so the authorities were concerned about those.
Okay, so when we talk about infectious diseases, okay, we said it's a virus, we said it's an RNA virus. Does anybody know what family it is? Paramyxovirus. What are other paramyxoviruses? Louder. Hendra? What else? Pass the microphone back.
testing your basic microbiology again.
In domestic animals, for the vascular
CBV, canine distemper. What's an important one in humans?
What's the one today that's been eradicated this year? What's the two diseases ever been eradicated from the world? Yeah, well, you were listening. Dominic mentioned this morning.
Rinderpest. Yes.
Rinderpest, yes, so rinderpest is in fact, just as an aside, some very good articles if you're interested in the history of infectious diseases that suggest that rinderpest or canine distemper virus may have caused disease in humans, or it may have been the progenitors of disease in humans. What disease would that be?
You're all vaccinated against it.
What's the other paramyxovirus of very high importance in humans? Measles, yes. And again, if you look up there's a very interesting historical article from Japan looking at the history of measles. It may have come about from contact with humans when they first domesticated either dogs or cattle. Nobody's very sure, but there's some debate about whether rinderpest became measles or canine distemper became measles centuries ago.
So when the reason that part of the reason that I am somewhat painfully making everybody go through that exercise is because just like when we study animals or when you're working with different animals, it's important to think about their taxonomy and who they are related to. Because if you're working with a cat or if you're working with a leopard, you have to think about domestic cats because that's what you're familiar with. Because usually when they're related, they're going to be similar. So it's with viruses, it doesn't always hold true. But if you think about all of those viruses, they're all very infectious viruses. It's very easy to transmit them. So it's when you think about that whole collection, that's why it's important to think about Nipah, that it's not something like Ebola or HIV or anything. It's one of these very easily transmitted viruses that fits in this paramyxovirus family. So when you're thinking about the ecology and epidemiology of infectious diseases, it's always helpful to have some idea of what families they belong to and what the closest related ones are, because that will help you to understand them. Okay, so we've talked about what kind of virus it is, what geographically where it is, starting with Malaysia. Is there anywhere else besides from Malaysia? Bangladesh. Can you name a country that's between Bangladesh and Malaysia? Thailand. Okay, we've talked about different animals that infect. So what's another important element when we're talking about infectious diseases? What's something else that you would want to know if it were?
How do you get it?
Yes. How is it transmitted? Yes. Transmission mode. What do we know about this virus? This family or this group of viruses? How are they transmitted? Think from breathing. So actually, we don't have to go into this a whole lot because this is a fairly new virus, and there's some things that are known, there are some things that are unknown, but basically it seems that you can get it from ingesting it, so you can get food contamination. It seems that you can get it directly from contact with fluids from animals, so from pigs, that's how many people have been infected. It also seems that people can transmit it back and forth between people, so there's probably a respiratory route. So again, this is part of the nature of a very infectious virus, so that once it's shed from your body, it's very easy to transmit it to other people. So, and that's an important thing to think about with this, or back to other animals as well. So I think we got most of this. So the paramyxovirus family. So Hendra and Nipah are very two similar viruses. The reservoir is believed to be this Pteropus genus of fruit bats. We didn't really talk too much about symptoms, but then again, many of these are still question marks because we're still learning, and this is something that's probably fairly new in the human population, and we don't know much about how it's affecting animals. But it seems that in bats there don't necessarily seem to be any symptoms, which is usually what's true of a reservoir, right? That's part of the reason that a reservoir can maintain a virus or a bacteria is because it doesn't really affect it very much, usually, not always. In pigs, it seems to be pretty mild. It seems to be mild respiratory disease, although I think there are cases where pigs also die from it. And in humans, it gets very severe. And again, this is a classic. It's about. This is.
You might want to slow for the translator.
Okay. This is a very classic picture of an emerging infectious disease. So something that is existing in wildlife, fairly undetected. And if you remember the graph that I showed yesterday with the curve, the waves of something happening in wildlife, and then every once in a while it spills over. So in this case with Nipah virus, sometimes it spills over into pigs, and then pigs can infect people. Sometimes it spills over directly into people. So in some ways this is almost a perfect picture of these diseases that we're worrying about that may become bigger and bigger problems with human populations.
About some of the factors. You've established that it's bats, pigs and humans principally. Although Bangladesh, why would you think that pigs might not be so much of a involvement in Bangladesh? This is social anthropology. They're Muslims, yeah. I'm told, funnily enough, that there are pigs in Bangladesh, small numbers of pigs. There are communities there that are allowed. It seems to be reasonably acceptable that some communities use pigs, but very low numbers. So the first time it came about in Bangladesh, people thought, wow, something quite different here. Just thinking about the clues. What close contact do bats have with pigs? The pigs and bats live in the bats roost in the pig pens. How does the disease in bats get into pigs? Bats and pigs share habitat. Okay, but you've got to think of it. We don't have time to go into it in great detail, but if you think about it, we're talking about bats and pigs share habitats. Did they always share a habitat when they built the pig farms there? Was there something, I think it was either Dom or Chris, talked about deforestation and change of habitat. And certainly in Australia with Hendra virus, which is very similar, it was related to cutting down a lot of trees, the bats having to go further and further afield to look for food, huge numbers of bats, and becoming closer and closer into human habitation, seeking out trees. So there's a lot of factors involved in these disease emergence.
I just want, and also we didn't go into a lot of detail here, but if you look at any of these, the websites for FAO or CDC or WHO, they're all good places to look for the general information. Usually in those places there will be a very short summary and also much more detail depending on how much detail you want. But you also have to be careful with fairly new diseases like this. That even if one of those websites is hasn't been updated in a year, there might be very different things that are known about it. So you always have to be careful with these things that are new and being more and more investigated to make sure that you're looking at the best information. So for me, I think about these in pictures. So I think I found slightly different cartoons than Dominic yesterday, but again, so the reservoir is bats. The solid lines are ways that we know the virus is transmitted, so into humans, into food or bananas, into pigs, also to some domestic animals like dogs. And there are question marks still about wildlife, and it seems pretty obvious that it should be there in different kinds of wildlife. But part of the reason, well, what is the reason that we don't know? What is the reason we don't know most things? Nobody. And it's not completely true. But for the most part, it's part of the reason that there is a question there is because there hasn't been good research yet. Nobody's looking very hard, and as Dominic pointed out this morning, this is one of these areas where if you're going to go looking, you have to be ready for both the answers. So this is a classic research situation where you have to think about the answers to the questions before you start to ask them. So if you're a wildlife veterinarian and you want to go start testing wild boars in your park, what are you going to do if you find them, if you find positives? And we'll get into some of this with the surveillance.
That picture is very, very important, I think, because it shows you that, if you've got a new emerging disease, I mean, this is only about a decade old, possibly more, but I think it's 99 or something it came about. You've got to think laterally, you've got to think sideways. And so you know that it caused disease in domestic pigs. Well, the closest relation to domestic pigs is wild pigs or feral pigs. So if you're looking for reservoirs or looking for evidence of disease in other areas, you go to the most closely related animals first, because that might be another wild animal that could be involved in the disease. And then you look at people that may be in contact with those as being at risk.
And just again, so a reminder, so this is a map, and the red area is the Hendra virus, so that's the closely related one. And the big part of the map, so that whole shape, that's the range of the bats from that region that are known to carry Hendra. So the specific red stars, those are where there are cases or outbreaks. I can't remember through which year, but it's obviously through at least 2006. So there have been reported cases or outbreaks there, but we know that the bats go all the way up into parts of Indonesia. And this is Nipah in the blue. So again, cases in Malaysia, or outbreaks in Malaysia, many in Bangladesh, but the bats are known to range all the way through here. So it's very likely that there's Nipah and bats, at least, in all of those places. And it's, since it's spilled over in Malaysia into people and pigs, and in Bangladesh to people and I think domestic animals, then it's likely that it's just waiting to come out everywhere else, including right there.
How many of you have fruit bats around the areas that you work in? Have you seen fruit bats? I have species of fruit bats in Thailand. That's only in some areas.
And do the people eat bats in Thailand?
No.
Yeah. And are they fruit bats and no?
They eat bats in Laos. In Laos they eat whatever bats they can find, but they're usually the ones that I've seen usually insectivorous bats, the small ones.
And I think that's my understanding too in this area. But again, and that's many of those things. There may be things that average people might eat as bushmeat, and then there might be things that poachers and other people might eat. So it's always, and especially now when you think about people moving back and forth between countries and displaced people. So if you have unrest in Laos, there might be refugees that come to other places. So just always something to be aware of, that even if it's not traditional to eat bats in Thailand, it could be something that starts to happen just because everywhere in the world is becoming more and more mixed. So it's just always something to think about and be aware of. Okay, but to talk about surveillance, I think just a bit of a reminder, and Dominic covered some of this morning, but again, when we think about monitoring programs, this is the practice of collecting, recording, summarizing, and disseminating information related to health and disease in the population in an ongoing manner. And usually we're talking about endemic things. So we all know what endemic means? Yes? Some nodding. Does everyone know? Okay.
Everyone knows endemic, but could you define it?
Yes? Okay. So, so in this case, with the situation that I presented, are we talking about an endemic disease? Anybody? So if you're working in a national park, say one of the ones here close to Bangkok, and you have a community outbreak of Nipah in humans, then what is your expectation for if you're doing wildlife, if you're asked to do wildlife investigation, and you have to classify it, thinking about what you're doing? So the big question is, do we consider that Nipah is endemic in wildlife in Thailand? No, right? It could be here, but right now nobody knows. So we would consider. So in this case, we wouldn't be monitoring wildlife for Nipah. So this would count as surveillance. So part of the idea, just like avian influenza surveillance, is we're trying to. It's basically like monitoring, but it's with the expectation that if you find something, it's bad and you need to do something. So it's not as if we're just monitoring a cycle of, rabies in domestic dogs in Bangkok or something like that. This is actually surveillance. So the idea is that we have something that we want to prevent from getting into the area that we're working, right? And also we distinguish this from an outbreak. And even though in what I've described, there is an outbreak in people. And we're going to talk much more about this tomorrow with outbreak scenarios, but if it was an outbreak in wildlife, then there would sort of be different things to think about. So let's just go forward thinking that you know that it's infected people locally, you're responsible for the wildlife, your boss has asked you what to do, and now you have to create some kind of a surveillance program, okay? So this is the first part of the surveillance program, is what is the objective? So who here can explain or articulate the objective in this case, the scenario that we presented? What is your boss's expectation? So you can't, I think you all say, yes, we need a surveillance program, but before you do that, we have to go and figure out what that means. So what is, in this case, what are we trying to do with the surveillance program? What is the objective?
You've told Chris that Nipah is not endemic here, so therefore it's exotic to Thailand. It may be on the government list of diseases that we don't have in the country, that we don't want in the country. So, as Chris said, what would be the first question you would want to answer?
Right. Is it present even? So even before the question of is it endemic, which means that it's fairly prevalent and it's maybe been here for a long time, just is it here at all? So especially if you have this scenario where you know that it's spreading amongst people, that means the virus is here somewhere. But what are the possibilities with people? If you have a community of people, it could be that somebody got it from wildlife here in Thailand and then they've infected other people. Or it could be that somebody came on an airplane from Malaysia or Bangladesh and infected other people. And each of those scenarios is very different. If you're the wildlife manager in a national park and you have people nearby that are infected, you should be very nervous, because either that means that they're getting it locally or somebody has brought it in. And you also have to worry potentially about if it's in people, then it might spread to wildlife. Because remember, when we talk about zoonotic diseases, they don't just go in one direction. So, Genetic diseases are those that are shared between people and other animals. So not necessarily just ones that people get from other animals, but we can also transmit them back the other way. Okay, so if that's our big objective, is to try to figure out if it's in wildlife, then what are some more specific objectives? And maybe we'll get into this more if we go through the design, but let's...
What we've established on the other page that there's 3 or 4 major animal species involved, or that have been involved in Nipah isolation. So, one of the thoughts, is Nipah present in Thailand? One of the thoughts would be, even just thinking about a surveillance program, is where you target it.
So I think part of what we're trying to get at is, so the question is it in Thailand? And then the next question is it in wildlife? And the next question is, what kinds of wildlife? So and we'll get there in a second, I think, with the design. But going back up to the way that Dominic laid out these questions earlier, if we're thinking about One Health integration, so if we're thinking about the environment and wildlife and humans, there's a question of who will use this data. So one of the things that you need to be asking yourself is, if we're going to do surveillance and start looking at wildlife, are we doing this so that we know about wildlife? Are we doing this so that public health and medical doctors can know about the risk to humans? And again, these are things that you may not have any control over, the answers to those questions, but they're things that you need to be aware of, again, before you start a surveillance program, because it may depend on how the data is going to be analyzed, how it's going to be interpreted, and what might be the actions that happen from those questions. Okay?
Just jump in again. This is why it is so important, why we laboured a little bit at the beginning, the natural history of the disease. You have a limited amount of money, the governments have a limited amount of money to put into surveillance programs. You have to know about a disease, or at least be able to predict, even with an unknown, a new disease, as to what might be the animals involved. You can't go for Nipah virus and do a surveillance program in rabbits or mice. I mean, you might, but you might waste an awful lot of money there. So you've got to understand the natural history to be able, and that's where the vets come in. And that's where the vets can work in with, your boss has asked you this question. Well, you would be able to think about, through reading the literature, talking to your colleagues in other countries, about what would be the specific area that you would target or put your dollars, put your baht into actually developing a surveillance program.
Okay? So this is the hard part. Or maybe not the hard part, depends on the situation. But so let's think about methodology in this situation. So what are we going to do to address this question of is Nipah in wildlife in your national park, in whichever one? I'm not familiar enough with them here to know which one, but whichever one you're working in, how are you going to answer this question?
Might even be other questions you might add into there. I think in this instance you could add in where. So where are you going to do your surveillance? We started to answer the who, what, and we talked about target species, because you can't just go and blanket sample everything. The where obviously is the area near the outbreak, I'd imagine, or at least the national park near the outbreak.
So with the surveillance, what are we going to look at? The who, what here refers to what's going to be the target of the surveillance. And this is an exercise. There aren't necessarily any right answers here. This could be done 100 different ways, actually, and some of those ways might be better than others, and you may not know which of those ways are better than others until it's all finished. So this is to get everybody thinking and talking and figuring out how to address these issues. So there aren't right answers here unless you said octopus.
And there's rarely been a surveillance program in history that people got it right first time as well, particularly with new emerging diseases where you have to think very fast. So making mistakes is not a problem, but you need to review regularly and update. Yep.
So and why bats? Aside from that we know that they're part of the system. So why would bats be a good target for surveillance? Okay, so that's I'm trying to think of so it's in this case that's one obvious target. It would be bats, because we know that they tend to be the reservoir and other places that we know there's viruses. So that's the most obvious target for surveillance. What are the pros and the cons of trying to get samples from bats, or trying to even target them, whether you're collecting samples, whether you're collecting observational data, whether you're just looking for dead bats, whatever you're doing, and we'll talk about that a little bit more. But what are some of the good things about using bats? Okay, one is that we know that they're probably the most likely animal that would have Nipah in the forest, right? Yes? It's an assumption, but it's a safe one. What's a bad thing about trying to target bats?
I'm thinking on my feet as well, because we haven't discussed this, but I can think of a couple of things. How easy is it to trap bats? Is it very simple? You just go out and catch a bat and bleed it? How safe is it to sample bats?
How many of you have done any bat trapping?
Oh, you. That's good.
Okay. So what do you know about doing that? Is it easy?
What's your experience? Talk about it, what?
So first, we need to know the source of in which region has bats, like the cave of the bats. In any region in Thailand, when we investigate, then we go there and we set the trap. Then we can get some, but it's not a huge amount of the bats. So we can get some. We can expect maybe 10, 20. It depends on time. Sometimes we can get less than 10, sometimes 10, may increase to 20.
Yes. As we said, the good thing about bats is they're going to be the most likely target. But bats are hard to catch. Sometimes depending on the species, there may not be very many. So not only are they hard to catch, but they can be hard to find, at least in good enough numbers. We know that bats are kind of dangerous. Yes, there's lots of diseases that, along with Nipah, that bats can harbor that are dangerous to people. So there's very high risk. Are there any other things? Okay, those are kind of the important things, but that's something you have to think about, because it might be that there is a trade-off. So let's move on. So okay, we've said that about bats. What are other animals we could think about doing surveillance on?
you do surveillance on humans? Yes? Why?
Like that, we know that people can receive their virus from the virus we can infect humans. So we should concern about human health. And we should do the survey, the zero survey or something like that on the human people who work associated or near the park or the park, the park ranger or some kind of people that work like that.
Okay. So does everybody agree that humans are also something that we should have surveillance on? So what are good things about sampling humans? What are some of the positives? What do we say with bats?
If I wanted 20 serum samples in this room, how long would it take me to get it? Between now and 2:30?
So just think of this. Humans are very easy to find. They're easy to trap. They're easy to sample. Generally, we don't have to worry about infectious disease risks, although I'm always handling blood. You have to be careful. What are some of the negative things about sampling humans, especially if you're a wildlife vet working in a national park? Yes, there are ethical questions, and I don't know about here in Thailand, but in most of the world, you wouldn't be allowed to do it. It's something that very often would be controlled by human public health authorities. So you may not simply be able to go out and get blood samples from people without the Ministry of Health or some sort of extra permission. So does anyone, what's the situation in Thailand? Have any of you ever had to do that or worked in situations like that? They do human sampling. Okay. So then in this situation, what are you going to do? Exactly. So, and again, this is part of what we said from the outset, that in a lot of these situations, and a lot of what we're hoping to prepare everybody for, is that when situations like this happen, that you're likely to be part of a team, and you'll be working with human doctors, you'll be working with the wildlife people that may be, that especially you've all moved to a new national park, or you're in a new area, so most likely you don't know very much about those animals. So there'll be wildlife people that have no health training, but they might be able to tell you where the bats are, where are the ones that are easy to catch, where there might be hunters that are going into the park that have interaction with those bats. So those will be people that you want on your team. Yes? So again, this is part of this whole idea of the picture of One Health. And here there could be environmental factors. As we talked about with Deepa, when you start cutting down forests, and that means that bats may not be living where they used to live. There may be pig farms that are on the edge of the forest. There may be bats have gone to roost in pig farms. And those are some of the environmental issues. So again, that's something else that needs to be included and to be considered to be part of your team. Okay, so we said bats and humans. What else could we sample? Wild pigs? Okay. So we're thinking wild pigs or domestic pigs? Okay. So I think aside from humans and bats, again, we're thinking mostly about wildlife, but to have the full picture, we do want to be thinking about domestic animals. And again, the good things. What are the good things about sampling domestic animals? Easy to find, something you're all very well trained in. You know what the disease risks are. You can do it legally. You have a system in place. You have a veterinary lab that knows how to take care of those samples. what are the negatives in this situation? What might be a negative thing about going into this community and sampling dogs and cats and pigs? Think about what your objective is. Say again? So it could be, there could be some sort of environmental contamination. But the bottom line is that even if you sample humans and domestic pigs and dogs, you may find that they all have Nipah virus. But when you go back to your director and he asks you, what's the answer to my question? What have you done about the Nipah virus in the national park? You still don't have an answer. You still don't know if it's there, or you still haven't looked at wildlife. So you may have a better picture, and it may be that if you know that it's in domestic pigs or dogs or humans, that means that it almost doesn't matter if it's in wildlife, but you still haven't answered. Your main objective in this exercise is to think about surveillance and trying to determine if Nipah is in the wildlife in your park. So looking at the domestic animals will help paint the whole picture, but it's not going to lead directly to your objective, right? Okay, so are there other wildlife that we could look at? What would be one obvious one that Stan mentioned and was in my picture? Wild pigs. So, and again, same sort of thing when you're thinking about wild pigs. What are the good things about sampling wild pigs? Has anybody here worked with wild pigs in Thailand? Yes? I saw you nod. So what would be good things about working with wild pigs? Nobody has anything. How many of you have worked with domestic pigs? Dominic? Thank you.
If you can make sure that one of wild pigs haven't contact with human animals, if they have zero contagion, you can make sure that it's not the zoonotic type is not from human to animals.
Okay, so that's an important part of the question eventually, if we do find it in wild pigs. But even just about thinking about it at the most basic level, if your director told you that he wanted you to do surveillance on wild pigs, what's your feeling? Would that be easy? Would it be hard? What are the benefits? What are the difficulties? Especially, so right now we have a choice. We've talked about humans. Hopefully that will be done by the human medical community. We've talked about bats. We've talked about some difficulties and some challenges. So now we want to think about something other than bats. So we're talking about pigs. So in comparison to bats, what's good about doing pigs? What's bad about doing pigs? How is it easier then? So her answer is that it's easier than sampling bats. So do we agree that it's easier than sampling bats? I see some nodding and some shaking. And both of those are right answers. I don't know. I've never chopped either one or worked with either one here in Thailand. But this is the kind of thing that as you're getting to be wildlife veterinarians in your parks, you're going to have to be able to have an understanding of what are the trade-offs. You know, it might be that it's very easy to find feral pigs and that they're easy to trap and they have known routes where you can find them. And again, as I talked about with the gorillas, this is why it's important that you know the basic ecology of these wild animals. So it could be that these wild pigs come in huge herds and you can make a big corral and catch 100 of them at a time. It could be that they use the same trails and you can set traps for them on the trails. It could be that they're hunted in some places and that you can work with the hunters. I mean, this is how lots of wildlife surveillance is done in the U.S. with animals that are hunted, and then you go after the hunters kill them and you take samples from them. And that's pretty easy. You let somebody else do all that finding and trapping, and then you just go and collect the samples. And I don't know the situation in Thailand, but if that's happening, that makes it pretty easy. The other trade-offs are feral pigs can be pretty dangerous. So whether they're easy or hard to trap and find, it can be very dangerous to work with. We know a little bit about diseases of swine, probably not as dangerous as bats, right? I mean, the animals themselves are as dangerous, but there aren't as many infectious diseases like rabies and those group of lyssaviruses and things like that in pigs. So there's some trade-offs, right? Okay, let's add one more animal that we can think about sampling. Does anybody think of something else that we might want to sample? So we talked about the fact that we know that Stan mentioned that it affects domestic dogs. So is there any wild equivalent of domestic dogs here in Thailand? Yes, and they're called? Yes. So there are a couple of types of wild canids that might be in your parks. Yes. So those might be something else that's a sample. And again, when you're thinking about the surveillance, you have to ask these same questions. What would be good about trying to sample dholes or jackals? Have any of you ever seen a dhole or a jackal in the wild? Yes? Okay. Easy to find, easy to trap, dangerous to work with? Yes.
It's very difficult to track it.
Yeah. So again, that's the kind of thing that you have to think about. But there's also, with some of those animals, you also have to think about opportunities. So it could be that they might be very difficult. They might be very difficult to trap. But if you have a scientist that's out doing that as part of another project, then again, if he's doing all the work, then there might be samples. You might be able to take advantage of that and do some of that sort of opportunistic sampling. And it's in some ways it would be very hard to have a complete picture and to look at. You may not be able to describe is there the prevalence of dholes in the whole population. But if you sample one dhole and you find that it's positive for Nipah, that's helpful for surveillance because then you know that there's at least one dhole in your park that has Nipah. So in some cases, usually when you're doing surveillance, you're trying to think about for the costs, both for time, for materials, for laboratory work, what's going to be the easiest way to answer the question. And so in this case, it could be that you need to go out and trap a couple hundred bats and do all those resources, and there's costs and benefits with that. You might have somebody that already is collecting blood samples from a few dholes. You could have hunters that are capturing pigs, so you may want to do things in a bunch of different ways. But let's make a decision here just to move forward. So of the choices, who wants to sample bats? Raise your hand.
If you want to sample bats.
You're going to have to vote for one of the three. So first for bats. We've got 3, 4, 5, 6, 7, 8, 9, 10. Okay, 11. Okay, who wants to sample feral swine, feral pigs? Dominic, he's going to vote for all of them, I think. And And who wants to sample wild canines, so does and jackals and those other animals? Nobody.
Not even the person who's done it before.
Okay, so.
That's it. Take a long time.
Okay, so we've decided that we're going to do bats, understanding the pros and cons. So the next question is how many? And we won't go into too much detail on this right now, but there are ways that with calculations from epidemiology that you can do, that if you can make an estimate of, you think that the prevalence is so high, and how might you do that? If you were going to try to do this calculation, and I told you one of the ways to do it was that you have to estimate what the prevalence is of bats and the animals that you're going to sample. How would you estimate that?
Think about the previous lecture. Just before lunch.
You could all do this right now. Yes. You look at the internet. Look at what's known. So there, you can either look at data from places like Malaysia and Bangladesh, where there have been lots of studies. But even I found a survey from here in Thailand looking at Pteropus bats and seroprevalence. So you look at that data, and it says, I can't even remember what the numbers were, but if you look at that data and say it's 10%, then you use that as a number to figure out, okay, if it's 10%, then how many bats do I need to catch to be able to detect that, and with confidence that I'm going to detect it. Because you don't want to do a surveillance program like this and trap 50 bats and say, yeah, we didn't find it, but we didn't trap enough to be able to know whether or not it's there. So this is a very important part of study design and doing power calculations and those sort of things. So we're not going to get too much into that right now, but that can all be done, and that's a very important thing. Because much too often, when surveillance programs are done, especially with wildlife, there might not be enough resources, and everybody's kind of lazy and doesn't think about, rather than designing it and doing it properly from the beginning, we very often say, you have your director that says, here, you can have $1,000 to do this, and then you do whatever you can. And maybe that's better than nothing, but the problem is that if it's not well designed, you may spend all those resources and not find anything, but not be very sure that it's not there. So this is something that's very important when you're doing these programs. And again, part of the reason that we're glossing over this, I know that you all want to know how to do this and do this proper study design, but as Dominic said this morning, we could spend all week going over this sort of a specific thing, and it becomes somewhat complicated. It depends on the issues, and there are good resources for that. So as I think our advice is that for you now, as wildlife veterinarians, or even in zoo situations when you're trying to do these things, if you don't have an epidemiology background to do it, you should at least know that there are people that have that expertise that can help you. And the important thing now is that you have an understanding of what it means to do those things, what the problems are, what the pros are, because it could be that you sit down with an epidemiologist and they say to you, yes, you need to go out and catch 5,000 bats. And hopefully your response is going to be, I can't catch 5,000 bats, because as the wildlife vet and working with the national parks people, how much money you have, how hard it is to catch those bats, and those sort of things. So again, this is part of being able to work with a team, to be able to understand the issues and the language, and to find help where you need it, and to give help where you can give it. Okay?
And this, just to add to that, this is where it's very important to understand again about the behavior of the animals you're dealing with, the ecology of the animals you're dealing with. Wildlife surveillance programs often have a degree of compromise in them. Compromise because, one, we don't have enough money to do what we'd like to do. It's not the same as going to a pig farm and taking 200 pig samples. It's quite straightforward. You can do it in half a day. The logistics of wildlife sampling, wildlife surveillance, are very, very high. Human resources, technical resources and financial resources. As Chris said, you've got to do it right. It's the most important thing. But you may not have enough money. So you may have to have, this is where we're starting to build up, as Chris is talking and you're talking, an idea of the kind of, even with Nipah virus, the kind of components of a One Health team. Now you may not all be sitting around a table working together, but I suspect in a new emerging disease, it would be quite easy to get these people together. You've got to have your vets, both government from DLD and the national parks, because they have different expertise, public health, wildlife ecologists and zoologists, to be able to tell you more about the life cycles, the life history, where to go, how to trap them. They may be doing that already. Environmental scientists, somebody mentioned. Pig hunters. If you're looking at feral pigs, these are the people, if it's legal to hunt pigs, they know where to go. They know how to trap them much better than we do. Get expertise from them. And epidemiologists. So you go back to your epidemiologists or your biostatisticians and you say, we cannot do 2,000 bats. It's technically impossible, and it would take us maybe 5 years to trap 2,000 bats. It's not very easy. How can we work around it? So they can compromise on the statistics to get something that is a valid sample, but is an appropriate sample.
Just to even take another step back, as we're sort of talking about that bigger issue, it could be, again, going back to the objectives and how this data is going to be used. If the real question is just is the virus in this area, and there's an agreement that there's probably nothing to do about it because once it's here, it may not matter. It could be that after you go through this exercise and think about the pros and the cons of doing a wildlife surveillance project, if there isn't anything that's going to be done. So, for example, if there may be economic issues usually that won't relate to wildlife, but if you're doing this in a pig farm, there might be questions about, okay, if you find it in a pig farm, they're going to kill they're going to slaughter all of the pigs to try to get rid of this. And this is often, with something like Nipah, this will be an important question because it may be that it's in pig farms already and that you can kill all the pigs, but if you have it in the wildlife, it's going to keep coming back to the pigs. Or it could be that it's only in the pigs and it's not in the wildlife. But if it doesn't really matter, and you just want to know that it's there, it could be that the surveillance is much better done just looking at domestic pigs. And in that case, a lot of the study design becomes much easier because then you can very easily determine, if you're on a pig farm, you can sample as many pigs as you want. It becomes much easier. And there might be more money because there's industry involved. And again, the sort of thing, when thinking about surveillance, you should always be, the most important question is why are you doing it? And what's going to be the result of that? So again, this is the very important thing that Dominic tried to make a point of this morning, is before you're going to tackle these issues and ask these questions, you have to know what is the response to the answer. And so that's something that we, as wildlife veterinarians, we can very often be caught in a fairly dangerous place because we may want to know if it's in the bats in this area, but if it's in the bats in this area, then it might be that they're next going to test all the pigs, and it could be that in your surrounding community is a province where there's a ton of pig farming, and they're going to slaughter all the pigs. And so these are just the issues that you have to be very aware of and always thinking about when you're asking these questions and doing these kind of surveillance. Okay, so then the next question, I think, and it's sort of important that we get at least to this one, yeah?
I think it's worth spending time on this. This is really, this is giving a sort of a background of surveillance thinking, if you like, surveillance methodology. We're not asking you specifically to design a program, but what Chris is trying to lead you through is the way of thinking before you set up your program. There's an awful lot of work to do before you actually go out and sample animals. In fact, sampling the animals is probably the easiest bit of the surveillance program. Getting it all set up properly and most appropriately is vital, absolutely essential. Just a very quick thing about...
Sorry, if I can just interrupt 1 second. We may need to make a choice between finishing this example as a group or splitting into the small groups and trying to work through a problem in small groups together. And I'm not sensing that either people are interested or in doing more of this, or if it's still just working on the getting into the brain, because it's been a lot of information. So I'm not sure what you prefer. Do you prefer the group approach to working together, or if you'd rather just go and have small groups working together? But we don't have time probably to do both at this point for the rest of today. We probably have to choose.
เดี๋ยวเราจะทำตัวอย่างนะคะดีไซน์ออกมาทีนี้อยากจะถามน้องว่าน้องอยากจะทำเป็นกลุ่มใหญ่หรือว่าแบ่งแยกย่อยเป็นกลุ่มเล็กค่ะใครอยากทำเป็นกลุ่มใหญ่ยกมือขึ้นไม่มีใครยกมือใครอยากแยกทำเป็นกลุ่มเล็กยกมือขึ้นเอ๊ะ
Do what? So, I think they're fed up with hearing our voices. Should we just
wrap this up then, just to show them?
Yeah, so I mean, the rest of this is just all of the elements that Dominic went through before. So the next question here would be, okay, we've decided that we're going to do bats. Are we going to do passive or active surveillance? Everybody remembers what that means? Okay, and then how often are we going to do it? And again, so I think what our idea with the small groups is that we'll basically get in and look at other specific infectious diseases that you're interested in and that are important in this area, and then kind of go through these things again with like a little bit more sort of strictly. But again, so think of so the important message that I want to leave you with from this exercise is to think about all of those issues, and that it's very easy in these situations to kind of jump ahead and say, oh, I want to do surveillance on macaques in my national park, but you should always be thinking about what question is that going to answer? Is that the easiest way to answer that question? What are the implications? And go from there. So I'm going to turn it over to Sam. I'll see you have a break scheduled.
Do we have a break?
Okay. I'm fine. How's everybody feeling? Get up and stretch. Does anybody have any questions? I think we've been talking back and forth, but are there questions or comments? The other thing is we want to make sure that you guys feel like you're getting stuff that you need and that's useful. So and it's we've looked at some of the results from the surveys, but it's been a challenge for us to figure out how to use these 4 days to make that are the most useful to you, so that you leave here this week feeling like you've learned something and that you've advanced your careers as wildlife vets, at least in terms of epidemiology. Questions or concerns or comments or anything? And don't be afraid to tell us if you've been talking too much, or we're too boring, or that you want more statistics.
Okay, we are ahead of ourselves in time because we had a sort of a 2-hour section scheduled for that last session, which is probably far too long. I think we probably covered plenty on that. And as Chris said, what he would have done, what we were trying to do was pick a disease that you all knew, which is, you did know a little bit about Nipah virus, but in some ways to show you maybe how much you don't know, and that's not a criticism. We don't know everything about anything. For example, I want to ask you, what is a dole? I don't know what a dole is. Can someone describe a dole to me, please?
Dole is one of 2 tiny in Thailand.
I've never seen a dog. So
It looks like
a chihuahua.
it's like the weight is about 15 kilograms. 15 kilograms, and the hair is brown. It looks like a dog, but have a woolly hair,
Many in Thailand?
and they live in a group or in a herd, or maybe sometimes to 50 individuals in a herd. And there are
Who's the dog expert?
can you explain?
I don't think you're not, but I mean, and how many would there be? Is it quite a rare animal?
Endangered, yes?
It's
Or is it somehow protected?
in Thailand.
He's going to look up Wikipedia and find their pictures.
I see from Wikipedia.
Thanks very much. Now, I'm just trying to illustrate, we don't know everything about anything, so don't ever think that when we're asking you questions and you don't know the answers, that there's something bad about that. That's unimportant. You're learning.
And I'll just add that the only reason I know anything about dolls is I never even heard of them until a few months ago, and one of my colleagues from Washington was coming, I think, here to Thailand to do some capturing. So that was the first time I knew anything about dolls. So I'm a few weeks ahead of Stan, but not any smarter.
I thought a doll was something that young people played with. Okay. What we're going to do now is we're going to get you to go through a similar exercise, but there's a couple of in yourselves in small groups. But there's a couple of steps that we need to go through. We've got time to sort of spend just a few minutes on each of those steps. And the first step that I thought we'd go through is that when we split you into small groups, we're going to give you you're going to choose, one, you're going to choose your groups, and two, you're going to choose your class of animal that you're going to look at. Okay? So what I thought I might do first of all is just write up on a flip chart, get you just collectively giving me some names of groups of animals, particularly ones that would be important in your workplaces. So in your areas that you work, or the national parks that you work. So the wildlife in particular. So what wildlife groupings, we're not talking domestic animals, what wildlife groupings can we group animals into in Thailand? General terms. We've heard a few of them already, so what would be can you think of some groups of animals? Shout out. Elephants, okay? How would you describe a group of elephants? What is the scientific term? They're herbivores? Sorry? Mega herbivores, okay. So we could have one group as herbivores. Which would also include, so we could split that into two groups if you like. Because that's one group here. Another group, what would be another group of herbivores, a general term? In your national parks, what are the sort of the herbivores you have? Sorry, Banting? So would you call them, they're bovids? They're bovids? And cervids, okay? Okay, so we could split those three groups, or we could put them together. We'll think about that in a minute. Okay, so herbivores. What other groups of animals would you have in your national parks? Carnivores. Okay, thank you.
Dole's going to be in that one.
Doles. Dole. I don't know whether that's the right way of spelling it. So doles, tigers, leopards, bears, jackals. Okay? Not that we need to, I'm not going to ask you to specifically comment on different animal species. We're just trying to get larger groups. But this is just important to let us know what's in Thailand. Okay, herbivores and carnivores. What other broad species groupings are there in your national parks? We're trying to lump things into disease risks. So we heard what's the Nipah virus. What was the animal species we've been discussing? Bats, okay? What other things are very, very common in the forest? Birds. What other things are very common in the forest? Rodents. What animal group are we missing? What are we?
Primate.
What are we?
Primate.
Primates. That's a good sound. I was going to sort of do a bit of a dance here, but no, I don't need to do that. Primates. Can you think of anything else that we're...
Things with scales.
Reptile.
Thank you. We've probably pretty much exhausted it there from the perspective we could go into things like amphibians and fish, but I think we'll just give those a miss for now, realising that they have their own diseases, they have their own possible zoonotic diseases that they can transmit to people. But I think in general terms, we'll work with those ones there. I think possibly what we'll do is we'll keep herbivores together in one group, and carnivores. And then, so the next thing is, we've got these 7 groups. Are there any other major groups that I've missed? Insects. Okay, insects was mentioned. As vectors of disease, and actually very good sentinel populations used for surveillance in many countries. Okay, what I thought we might do now is we've got 8 groups here. So if we split ourselves, or at least the group here who are here to learn, into 8 different groups. So how many people, what's the numbers, how many people we have? 21. So groups of 3 and a couple of groups of 2, or we could actually tell you what, if we leave insects out for the moment, and you can think about insect-borne diseases. So we've got 7 groups and 21 people. So you could split yourselves in a minute into groups of 3. It's a nice, easy number, okay? Now you can split yourselves into groups of 3, so we get 3 people in each, and you can come up and call yourselves, or write down in blue the group that you want to work with. Now there's probably going to be a free-for-all run here, but I don't want you all to fall over yourselves trying to put up your names here. We could do it by vote, or we could draw letters out of a hat or something like that, but I think we'll just get you to choose a group. So you'll choose a group, and then you'll go away, and what I would like you to do is to come up with a list, and again, as I've said to you, don't worry if it's not comprehensive, if you don't know everything, but come up with a group of zoonotic diseases related to that particular animal species, knowing that there will be crossovers, because many of them are non-group specific, some are group specific. You can think of also emerging diseases of wildlife themselves for that particular group. So you can come up with just some thoughts on the types of diseases that are of relevance or importance to you in your work situation. So zoonotic diseases and how we call it, emerging diseases of wildlife. Forgive my shorthand there. So you would go away and you'll brainstorm and come up with a list of these, and for each of those diseases, you'll try and come up with 2, 3 key epidemiological factors. Okay? So you're going to come back and say to me that disease X is transmitted in this way, these are the reservoir species, and it's usually found in this part of a habitat. So just some key features of the epidemiology of those. Nothing too difficult. Then, because this is about surveillance, that's the background information, if you like, that you'd like to know. And then what you're going to do is you're going to choose one, and if you're smart enough and you've got enough time, you want to do 2, you can do 2, but if one per group anyway, is pick one of the diseases that you think are important, and really just follow through that protocol that Dominic gave you this morning and that Chris went through this afternoon, and look at how you would design a surveillance program for that particular disease. So I'm not going to give you an example because it might just it might be the only disease in that particular animal species. But if you, yep, Chris Montgomery. Okay. That's a better idea. Okay. Sorry? Okay. What we can do, we can divide you into 7 groups, and you make a list of diseases, and you choose one of them to design very simply or answer some of those questions and say where you can't answer, where you need more information for the others. Or we can come up with a list of diseases in half an hour, come back, list those diseases, discuss them for a little bit, choose the 4 most important diseases, and then split you into 4 groups, and you go and design a surveillance system. Which would you rather have? Which way would you rather do it?
And part of it is that our original intention was to sort of break into 4 or 5 groups so that...
You understand? Yeah? Ron, do you understand what I'm saying?
Yeah.
Yeah? Can you explain it to them, please? Just in case I've got it wrong. A couple of them that didn't understand. So we can do it 2 ways. We can either do it that we don't see you again for the next hour and a half or hour, and then you come back with a list of diseases and a surveillance system for one of them, or at least an attempt at developing a surveillance plan, or we can do it in 2 stages. The second part. This part here is obvious. We're going to do a list of zoonotic diseases emerging and choose one. Okay, I'll go.
โอเคเข้าใจไหมคะน้องแบ่งเป็น7 กลุ่มนะคะ แล้วก็เรียกว่ามาดูว่าในกลุ่มนั้นเนี่ยมีโรคอะไรที่มันสัมพันธ์กันนะคะเริ่มมา1 โรคในกลุ่มนั้นlist of diseases ก็คือเรื่องมาว่าในโรคในสัตว์ของกลุ่มนั้นเนี่ยมีโรคอะไรบ้างที่มันสามารถเกิดขึ้นได้นะคะแล้วก็ออกแบบระบบการเฝ้าระวังของสัตว์ชนิดนั้นนะคะ
They're making a list of diseases in groups of 3. Diseases in groups of 3, yeah. No.
อันนี้เข้าใจไหมคะคืออันแรกก็คืออันนี้คือเหมือนกันนะคะคือว่าก็คือแบ่งกลุ่มนะคะกลุ่มละ 3 ลุ่มละ3แล้วก็ลิสต์มาว่าในสัตว์กลุ่มนั้นเนี่ยมันมีโรคอะไรบ้างที่เป็นไปได้เสร็จแล้วก็ออกแบบรูปแบบการเข้ามาว่าถ้าเกิดเราเจอโรคนี้ในสัตว์นี้เราจะออกแบบยังไงนะคะซึ่งอันนี้2อันนี้ก็เหมือนกันแล้วก็พอหลังจากคุยกันแล้วดิสคัสกันแล้วก็กลับมาแล้วก็มาคุยกันว่าน้องออกแบบว่ายังไงบ้างที่พูดอะไรผิดไหมเพิ่มเติมเลยค่ะ
Which was just only one disease, or all of them? For a surveillance system, just one disease. So what we could end up is, if we choose option 1, we could end up with a surveillance system for 7 diseases, there's 7 groups, or we could just come back and discuss amongst ourselves which are the 4 most important and split again into 4 groups. You like that idea?
The other thing I would point out that our original idea was to have 4 groups so that Dan and Dominic and myself and Chuck can review each group to help.
ก็พอเดี๋ยวเรากลับมาเราก็แยก So when we come back, we're going to bring out the 4 group,
What
right?
we're going to ask you to do now is this bit here, front A.
Oh, okay.
Okay, split you into 7 groups, 3 people, and you'll go away and come back with,
So we have to write it.
you choose one of those options there, and you'll go away and come back just with a general list of the diseases you think are important for that group. Then we'll discuss them again as a group and come up with 4 that we think are the most important for DNP vets in Thailand, and then we'll go away and design a surveillance system.
And then they can use it and choose one of the
Okay, so if you choose yourselves into groups of 3. This is the first group of 3. 3 people come up here and we'll pick you out.
You got the elephant guys. We need one more? You have 2 or? You need one more elephant person.
Either those 2 working on one of those diseases, or they come back and then we choose 4 of those 7.
คือเขาให้เราเลือก2 ช้อยนะง่ายๆมี7 3 21 3เนี่ยตลอดเลย7 กลุ่มตลอดไม่เลิกไปนะไปเลือกไปเลือกโลกชนิดหนึ่งเซอร์เวอร์ซิสเต็มมันไปออกแบบแล้วก็ทำงาน7 กลุ่มก็มาทำงานอย่างนั้นตลอดนั่นก็คืออยู่กันตลอดหรือ2 ข้อ B ก็ไป7 กลุ่มแล้วกลับมาเอา7 กลุ่มมาย่อยเอาเฉพาะ4ที่สำคัญถ้าเราคิดว่าเราอยากจะทำ4แล้วก็ทางนี้ก็แยกกลุ่ม4เพราะนั้นก็คือ7 หรือว่า7 4จะเอาแบบไหนจะเอา4 7หรือจะเอา7 4เราจะไม่ตอบเขาเลย
แล้วทำไมไม่ปัญหาเขามีอินสตรัคเตอร์ไม่
อันนั้นไม่สำคัญหรอกเพราะว่าเขา
แต่เป็นเด็ก
เดิน
ตั้งแต่แรกงั้นเราไม่ต้องทำงานต้องสอนไม่ต้องมีการสอนกับ
ก็ต้องเสนอเขาสิแล้วก็เสนอครับ
แล้วเราแต่เราจะขยายได้ไม่ได้ให้ดูเห็นแล้วได้ไงเนี่ย
So the lesson she is recommended, since you only have 4 trainers here, why don't we start it out as a 4 and then instead of 7 or 7-4, why don't 4 in the beginning?
Okay,
to begin with.
The only reason for starting at 7 because we came up with 7 groups now.
เหตุผลว่าเราไปบอกว่ามี7 animal ไว้เราอยู่ในป่านะ
That's the reason for that. Otherwise, we could choose the 4 most important groups and I would have
เขาก็เลยให้7เพราะนั้นเราก็น่าจะเป็น7 4มากกว่า7 7เพราะว่าจะมีคนเยอะด้วย
Let's just take a vote. Who would like to split into groups of 4? Groups of 6? Sorry. Who would like to split into groups of 3? And who would like to split the class into 4 pieces?
โอเคอ่ะกลุ่มออฟทรียกมือขึ้นค่ะกลุ่มออฟเซเว่นยกมือขึ้น
กลุ่ม4 กลุ่ม
กลุ่มที่มี4 กลุ่มอะค่ะ7 คนใช่ไหม
จะเอากลุ่ม 3หรือจะเอากลุ่ม 5จะเอากลุ่ม 3หรือจะเอากลุ่ม
กลุ่ม 5 หรือว่ากลุ่ม 3 กลุ่ม 5 โอเคคลาส
Okay, so let's have groups of 3 groups of 5, 1 group of 6, okay? So the first group of 5 people, you come here and choose an animal species. 5 people.
โอเค
5 คนนะคะ so one clinic vet and then for ENT vet right or just the matter okay so 5 คนนะคะมาที่นี่เพราะว่าตอนคุยกันเมื่อวาน5 คนมาที่นี่แล้วก็อีก2 กลุ่ม another 2
You're
group
going to make us do a little game. When I say 1, 2, 3, you will go into the 4 corners of the room here with your groups, okay? And you can change. Okay. Ready? I want one group of 5 in that corner, 1 in this corner, 1 in this corner, 1 in this corner. 1, 2, 3.